on, and they're still like snoring out there, and I'm like, come on, give me something. Give me something, huh?
And to think I almost
Yeah, cross bridging. Okay, so let me give you a preview of things to come. And so your test is going to be a week from Wednesday, yes? Okay, and it will cover synapses, muscle, and autonomic. We will finish muscle next time. Muscle is 2 lectures, autonomic is 1. But that means that you guys will feel rushed with autonomic nervous system unless you study it over the weekend before class. Yes? And there's no reason you can't do that. You can go through all of the notes, and you can even look at the lectures from last semester. It's really not hard material, but it's super wordy, so you need to do whatever you can to get the terminology down, and then you'll be totally cool with the physiology. Does that make sense?
So it's going to be on synapses and what?
Synapses, muscles, autonomic nervous system. Autonomic nervous system is just a 1-day lecture, so we're on track. I just don't want you to, from Monday to Wednesday, feel too rushed about that autonomic nervous system lecture. That make sense? Okay, so we're going to do muscle today. And the muscle today that we're doing today will be connecting with the synapses that we did last, yes? And assuming that you did have lab last week, it will connect with the lab that you did last week. So this is going to sort of put all of them together. We won't get into the connecting with lab until next time, but today's all about connecting muscle activity with synapses and talking about how it works. Okay, so are there any of you guys who either haven't had anatomy or aren't currently taking anatomy? Anybody? Okay, so that will mean that the anatomy that I'm going through today is going to be a little fast for you, so go back to interactive physiology and brush it off if you have trouble with it. For the rest of you guys, I'm going to go relatively quickly through the microscopic anatomy so that we can get down to the sarcomere level and then start wiggling the sarcomere, okay? But since I didn't have all of you guys for anatomy, I want to make sure that we are starting at least somewhere close to the same page. Okay, so as far as the functionality of skeletal muscle before we get into the detail, it doesn't just wiggle you, right? Sometimes skeletal muscle is about holding you still. It's also responsible for like postural stability, and like the skeletal muscle in your pelvic floor keeps your junk up there. So sometimes it's supporting things. Sometimes it's heat production, and we'll talk about how heat production occurs. Sometimes it's sphincters, like guarding openings. So it's not just wiggling, okay? But we'll start out with wiggling as the basic. Okay, so remember what we were talking about last time is we were talking about the fact that skeletal muscle is both highly vascular, remember? Highly vascular, it's got capillaries snuggling around every single cell because it's really, really energetically expensive. And then we also said that skeletal muscle is also highly innervated, meaning that how many skeletal muscle fibers need to be innervated for the whole muscle to contract? All of them. So every single muscle fiber, and there could be thousands, has to be innervated, meaning that, remember we said if you cut this one or interrupted synaptic transmission with that one, this muscle fiber would be paralyzed. So it's highly vascular and highly innervated. So let's get to the whole innervation idea now, and we'll get into it in some more detail later. This site at which the neuron communicates with the skeletal muscle fiber would be called a synapse if it was neuron to neuron. What's it called when it's neuron to muscle? Neuromuscular junction, exactly. Okay, now specifically, do you guys remember what kind of motor neuron is responsible for stimulating skeletal muscle? What kind of motor neuron stimulates skeletal muscle? You got it, I heard it. Somatic, yes, somatic motor neuron, right? So I'm trying to pull the terminology that you've learned but haven't like dusted off in just a little while. So this is a somatic motor neuron. Okay, and skeletal muscle contraction is generally, it's mostly voluntary with an asterisk, meaning that your diaphragm is voluntary, yes, because it's skeletal muscle, but you can't hold your breath to death, right? Even if you decide you're going to throw a fit and hold your breath to death, no, your brain will make you lay down and it will take over for you. So it's mostly voluntary, but not completely. Okay, so now let's get into skeletal muscle fibers. So please remember that when you talk about the word muscle fiber, you are using it interchangeably with the word muscle cell, right? A muscle cell is just so long and skinny, up to a foot long, in the longest muscle in the body, which is the sartorius, yeah? A cell can be up to a foot long. Since the cell is so very long, you use the word fiber to describe it. But is it a cell? Yes. It still has a cell membrane, it still has cytoplasm, it still has organelles, it still has all of that business. But we can acknowledge this does not look a whole hell of a lot like the cells you learned in 1610, correct? Primarily because of its unique functionality. Its unique functionality is due to these little Vienna sausage looking things inside, and those are called myofibrils, and we're going to get deep into their functionality today. But before we do, let's talk about the things that the cell has in common with all cells. So, some yahoo, I do not know why, decided that they had to rename every damn thing inside a skeletal muscle fiber. So you already learned plasma membrane. No, you have to call it the sarcolemma. You already learned cytoplasm or intracellular fluid. No, you have to call it the sarcoplasm now. So they renamed everything, and I'm sorry that you have to learn them, but you do because that's what they call them throughout the rest of your career. But a skeletal muscle fiber has similarities to other eukaryotic cells. So it's got a cell membrane, but now we're going to call it a sarcolemma. Okay? It is still a phospholipid bilayer with protein stuck in it and all of that business. It is going to have nuclei. Now, do you guys remember that skeletal muscle fibers are multinucleated? Yeah? This one is only showing you one, but it generally has multiple nuclei. And do you remember that the nuclei are also peripherally located, meaning they're usually pushed up against the sides? And the reason that they are is, can you see that the middle of the cell is kind of busy doing other stuff, right? So the nucleus gets pushed up against the side. So, other things. Notice that if you look at this thing, it has a disproportionately large number of mitochondria, which will tell you something about the activity of that tissue. You see those little pink guys? Those are mitochondria. So compared to most tissues, this has way more mitochondria than you're expected to see. And then a couple of things that are special about the plasma membrane before we go any further. So the plasma membrane, also called the sarcolemma, forms the boundary or the cell membrane. But in addition to forming the boundary, please note that if you look at the sarcolemma, it looks like it has little pores. Do you see that? Those little pores right there? What those are is they're actually the entrance to a tunnel which leads into the cell. So look here. What? So right here, do you see? Okay, so this is the cell membrane, and then it folds. And what it will do is that is a fold of the cell membrane that allows you to continue the cell membrane deep into the cell, which is going to be important because now if I have an action potential along the surface, it doesn't have to stay on the surface. I can actually take the action potential and route it toward the middle of the cell. Okay, those little tunnels that lead toward the middle in green there are called T-tubules or transverse tubules. Okay, and they are a continuation of the cell membrane, meaning that if the cell membrane had voltage-gated sodium channels on it for an action potential, so would the T-tubules, right? So you can take that action potential deep into the cell. Does that make sense to you guys? Okay, all right. So now let's look inside and see some structures that we're going to see inside. Okay, so the intracellular fluid in here is called sarcoplasm, and no, you can't see very much of it, right? Because it's mostly chock full of stuff, but there is fluid in there. And in the sarcoplasm, of course, you got your nuclei and your mitochondria. And you've also got some things that aren't labeled on here, but are physiologically important. One of them is that inside skeletal muscle fibers, each skeletal muscle fiber has its own little piggy bank of glycogen. And glycogen is stored what? Glucose. It's stored glucose. So it's got its own little piggy bank, and keep that in mind for just a second. And then in addition, inside here, also not shown on the picture but physiologically important, is that skeletal muscle fibers have a protein in their sarcoplasm that's called myoglobin. And myoglobin is similar to hemoglobin. Hemoglobin binds to and carries what around in the bloodstream? Oxygen. Myoglobin binds to and stores small quantities of oxygen inside each skeletal muscle fiber. So look what the skeletal muscle fiber has done for itself. It has stored glucose. It has stored oxygen. And it has boatloads of mitochondria. Do you see what it's doing? It's making sure that it can always do what?
Make ATP.
Yeah, it can always do cellular respiration, right? even if, for instance, I haven't eaten for 18 hours, can my skeletal muscle fibers still get me out of the way of a bus? Yes. It's your emergency response tissue. It can also do a little bit, always do a little bit of aerobic cellular respiration. Does that make sense? Now, why not depend on the glucose that's stored in the liver as glycogen and the oxygen that you're carrying around in the bloodstream? Because if a bus comes at me, right, do I have time to wait for glycogen to break down in the liver and the oxygen to be delivered by the bloodstream? No. So instead of going to the bank for the money, I have a piggy bank, right, in every single cell, and I don't share with anybody else. The cell is just for itself. Does that make sense? Okay. So inside the cell, you store all of that. Okay, and then,
Question.
yeah.
So, the sarcoplasm, is it in between the mitochondria, or is
It's
it
like anything that
all
you
of
see inside the cell that is not a physical structure would be where the sarcoplasm would be.
okay? So
It would be
not
the fluid.
not the little.
No, I'm going to talk about those in just a second. So anything that is not like a structural entity inside would be filled with intracellular fluid or sarcoplasm. Does that make sense? Okay, so now let's talk about these guys. So what the heck are these guys? These weird looking little things that make it look unlike any other cell. What those are is big Vienna sausage looking bundles of proteins. Okay, and what they do is this big Vienna sausage looking bundle of protein. Think about it like a coaxial cable. It's like a wire made of wires, right? And you can see that primarily it's made of those orange wires and the purple or blue ones, right? Okay. What that is going to do, the entire thing is going to be called a myofibril. And the myofibril will attach to the cytoskeleton at the end of the muscle cell or muscle fiber, right? And that is going to be the thing that ultimately contracts. And when that contracts, you can yank on the cytoskeleton and you can shorten the muscle cell. And we'll get into the mechanics of how that works today. Does that make sense? So the more of those you have, the more tensile strength you can generate. Does that make sense? So if it's a contractile protein and you have more contractile proteins, you can actually have a stronger contraction. So we'll get to strength of contraction and different size muscle fibers and increasing muscle strength next time. Okay? Go ahead.
So the more you have,
Not the more
So-
cells.
so someone that's really skinny and then they start lifting weight and they get a little bubble.
Mhm.
So how is it just
There,
You don't
I'm
get
going to
more of those.
oversimplify and we'll do it in more detail next time. But what generally can happen is if I start using that muscle cell
right?
and I start doing weight-bearing activity, which means trying to increase muscle mass. I'm doing high weight, low rep, right? I'm trying to increase muscle mass. What happens is when you strain that and then rest it and provide it with protein and also provide it with ATP, it will undergo protein synthesis and make more myofibrils. And then your muscle gets bigger. It's called muscle hypertrophy. Yeah, and we'll talk about the mechanics of that next time. That makes sense? Okay, so now one more player that we're going to worry about. So this is a myofibril. We're going to get into it in detail, but please note that every single myofibril has a completely snuggled all the way around with that purple structure. Do you see it? That purple net all the way around it? That is called the sarcoplasmic reticulum. And what it is the muscle's version of smooth ER, smooth endoplasmic reticulum. Does anybody remember what the smooth ER does in most cells? Anybody remember?
It's the transport.
That one is, that's Golgi. Anybody remember? For some reason, nobody ever remembers what smooth ER does.
I thought it like this lysosomes.
It's lipids. It does lipid synthesis primarily, but this one has a special function. Okay. In addition to the normal smooth ER functions for a normal cell, it has one extra function, and that is at this location right here. See that location right there, right next to the T-tubule? It stores calcium ions, and that's going to be an important player in our story that we tell this time. Okay. So most smooth ER does not store calcium. This one does. Okay. So we've got all of our players, and then we're going to start like making it wiggle and start talking about the functionality. But first, we're going to take this guy right here, the myofibril, and we're going to break it down. Okay. and we're going to break it down so that we can see the components, the major components of the myofibril. So look, and please acknowledge before I break this down that this is a three-dimensional structure that I'm going to represent two-dimensionally on the board. It's going to end up looking like, where are you? This. It's going to end up looking like that, but it's actually 3D structure. So I'm going to build it for you. And then we're gonna wiggle it. So if you've had anatomy, how many of you guys, anybody not go down to the sarcomere level when they did this in anatomy? Everybody go down to the sarcomere level? Okay. So generally speaking, people have had this before. Does anybody see a black one? A black pen? Get it someplace very special. Oh, there it is. Okay, so do you see, let's go back here for just a second. Do you see where this pattern starts to repeat itself? Jagged line to jagged line to jagged line. Do you see that? Okay. What I'm going to do is I'm going to flatten out one of those from jagged line to jagged line and build it so that you can understand the major proteins. And then I'll start to wiggle it. Okay. So this repeating pattern that you see repeated along the length of a sarcomere, along the length of a myofibril is called a sarcomere. Sarcomere. All right. So if you think of the myofibril like a train, a sarcomere is a boxcar, right? Boxcars on a train. Okay, so we're going to build one of those, and we're just going to do the major players first, and then we'll add some more detail afterward. So the sarcomere, the major players are these two proteins. And the proteins, of course, would have all been made on the ribosomes inside the cell. The protein that extends from the jagged line toward the center is going to be the thinner of the two proteins. So the boundaries are called Z-lines, or your anatomy book calls them Z-discs. It's the same thing, it doesn't really matter. So Z-line to Z-line is a sarcomere. And then there's two major players. This purple or blue protein that you've got right here is called, it's made of the protein actin. And it is the thin myofilament. Okay, so actin is the protein that's made of, we'll get into its structure in just a second. It is the thinner of the two, okay? And myo means muscle, right? And filament means long skinny, right? And it is a protein. Okay, and then the other protein that's the big player that you have right here is the thicker of the two. And it kind of hangs out in the middle. And this one is made of the protein called myosin. And it is the thicker of the two. The reason I put myo in parentheses is that sometimes they call it the thin filament and sometimes they call it the thin myofilament. Myo just means muscle, so don't let it confuse you if they add it or take it away. Okay, so why the heck do we care about this? Let's add just a couple players in here, and then I'll give you a preview of things to come. The couple players that I'm going to add in here is that the actin protein, if we look at it more closely, we will see that actin, actually let's look at this picture back here. Actin has these little dots, those little black dots that you see on there. I'm not going to represent them as black dots. I'm going to represent them as something that looks like handles so that you can sort of get the metaphor. And these are called actin active sites. And then the last thing to give you a preview of what's to come is what the myosin looks like when you look at it up close. The myosin has little projections sticking up off of it. And these are called myosin heads. So, a preview of the complexity that we're about to break down further than this. The way that I cause contraction is this. When the myosin heads reach up and grab the actin active sites and pull, what happens to the distance between the Z lines? Yeah, it shrinks. And if you shrink enough boxcars, you can shrink the whole damn train. And if you shrink enough of the trains, you can shrink the whole cell, right? And if you shorten enough of the cells, you can shorten the whole fascicle. Remember fascicles? And if you shorten enough fascicles, you can generate enough strength to move bone. Does that make sense? Okay. So this all starts at this level, which is why, even though students don't like it, you have to go down to the submicroscopic level before you can actually talk about the physiology of muscle contraction, because it all starts right down here. Is that okay? Okay. So let's just look at what this has to do with striations, and then we'll get back to the real phys. Okay. So we know that skeletal muscle is striated, right? Okay. So if you look at skeletal muscle, and here's a fiber, and the striations run perpendicular to the fibers, right? And they are light and dark stripes. Yes? Do you see that? Okay, so I want to know what this has to do with that. And I'm going to tell you that if you use a protein staining technique, then wherever you have more protein, it will stain darker, and wherever you have less protein, it will stain lighter, correct? Okay, so with that in mind, can you see that if you stain this thing, the darkest area should be where you have the thick filaments, correct? This area from here to here is going to be the dark stripe, which is called the A band. Okay, and lighter than that, always, will be from the edge of this A band to the edge of the next one, which isn't shown here, right? Do you see? Yes? Okay, so the next one will be the lighter stripe, which is going to be the I band. And the I band will basically go to the edge of the next A band. Okay? So with that, I'm going to have light stripe, dark stripe, light stripe, dark stripe, all along the length of the skeletal muscle fiber. YA and I, you don't really care because the names don't make sense to you, but they stand for anisotropic and isotropic. But here's a way to remember. A is the second letter of dark, D-A-R-K. I is the second letter of light, L-I-G-H-T. Okay, so the I band and the A band. Now, there is one little area that's slightly different, and you can see it on this figure. You can't see it on most light microscopy. So, do you see? This is a different type of microscopy, TEM, transmission electron microscopy. And so this is showing you A band, right, from here to here, and then I band. Do you see that there's a little area in the middle of the A band that's not quite as dark? Do you see that? It would be right here, where I have protein, but not as much because it's not overlapping A and I. Do you see that? Actin and myosin. So that little region right smack in the middle, right here, of the A band that's dark but not as dark is called the H zone. Okay. Why the hell do you care about all of this? Because long before we had the capacity to label these proteins, what we saw is, I'm pretty sure I know what's happening when I stimulate this, because when I stimulate the muscle, what happens is the banding pattern changes. Because you guys think about this. If I stimulate it, and it works the way that we said it did, the hands reach up and grab the handles and pull, right? When this sarcomere contracts, which one of the three of these, A band, I band, or H zone, will not change length? So think about it for a second. What?
It's an army question. I can't remember
Yeah.
what it is.
When you stimulate this thing, okay, if it works the way that we said it did, hands reach up and grab the handles and pull, okay, and then the Z lines move closer together, which one will not change length upon contraction? A band, I band, H zone. A band. It's the A band that doesn't change length. The A band basically pulls everything into it. So here is a picture showing you the A band and a relaxed muscle sarcomere. Do you see it? A band and contracted. Look at the distance of the H zone when you relax versus contract. And look at the I band, right? So interestingly, part of how they discovered this is that you can actually take a living muscle, and if you can get a thin enough slice and stick it under the microscope, you can stimulate it with an electrode and watch it contract. And so when I took physiology, we still used live animals in lab. So we took a frog gastrocnemius muscle and we stretched it over a light microscope. And then we stimulated it. The frog was still alive, but he was like anesthetized. And we stimulated it. And what you see is the tissue goes from looking striped to looking almost completely dark. To looking striped to looking almost completely dark. Striped, dark. Striped, dark. And so long before they ever had the ability to stain actin and myosin so they looked different, they looked at the pattern that was emerging under the microscope and they're like, oh, what's happening is these things seem to be sliding past one another. Does that make sense? Okay, so that's the basis of the banding pattern. It's also going to be the basis of the contraction. So let's go and talk about one more term and then we'll get into the details of this. When the hand reaches up and grabs the handle and attaches like that, okay, that's called a cross bridge. And we're going to be talking about cross bridges right now. Okay, so most of that you learned in anatomy, but now the phys that you didn't. Okay, so I want to talk about the way to make this contract. And then we'll go in and add some ATP to talk about the energy for it to contract. So we know that this thing is called a sarcomere, right? And the basis of sarcomere contraction is the myosin heads will reach up and grab the actin active sites and they are going to pull. When the hand and the handle, or the active site and the myosin head are bound to one another, it's called a cross bridge. So I want to look at a cross bridge cycle. Okay, before we do that, so this is going to be a cross bridge cycle, so look with me for just a second. Okay, we are going to follow one cross bridge all the way around, but I need you to understand and appreciate that there will be tons of cross bridges, but I'm following one. I'm following the cycling on a single cross bridge that will occur with one of those little, what do they look like? I don't know what the hell they look like. One of these little guys. This is going to form a single cross bridge. So what it's going to do is it's going to cycle. So let me give you a preview of cross bridge cycling. Here is what's going to occur. So this is what I kind of implied occurred. Hands reach up and grab the handles and pull, and it actually shortens the distance between the Z lines, but here's how it really occurs. What happens is the A band is staying in position, right? And I'm just following two cross bridges, one on each side. This one's going to go. So this one right here is going to reach up and grab an active site and pull, and then it's going to reach up and grab the next one and pull. So what it's doing is not sliding it close, it's ratcheting it. Does that make sense? So, you know the monkey bars that you did when you were a kid? Imagine that you are a badass beast of a child, and instead of going to the end of the monkey bar, you are going to stay stationary, and you are going to pull the end of the monkey bar toward you. Right? Does that make sense? You're staying in one position, and you're like, right? And you pull it all the way in. Is that okay? Okay, so that's what's going to be happening. But right now we're going to follow one cross bridge, one single myosin head, and see how it reaches up, grabs, pulls, releases, grabs the next one. Okay, so that's going to be called a cross bridge cycle. So, and I've got an animation for you after we go through it once and then add the ATP. So, it's a circle, so you can start arbitrarily anywhere, but I'm going to start the place that makes sense to me and the place that your textbook starts, which is forming the cross bridge. Okay, so cross bridges want to form. As a matter of fact, the only reason a cross bridge wouldn't form is a player that I'll put in just a minute. These are the active sites. Under most conditions, they are blocked, so you can't get at them. As soon as you get that protein tropomyosin out of the way, they will naturally form a cross bridge. We'll go back in and add the blocker in just a second, but it's out of the way. Okay, so I get this cross bridge formation for free because the myosin head loves to have a chemical bond with the actin active site. Okay, it wants to anyway. So it is going to reach up and it's going to form an association and a bond with the first one, the closest one. Okay, so that is free. Okay, it just naturally occurs. And here's what happens next. Next it is going to do what we call the power stroke, and the power stroke is going to do this. Okay. And then now it's slightly shorter. You guys with me? But now I'm stuck right here. Okay. Now what I have to do is I have to release, reposition so that I can grab the next one. Does that make sense? So one more time, cross bridge formation, power stroke, cross bridge detachment, reposition, cross bridge formation. Got it? Okay, so now what we have to do is we have to add in how ATP is used in this story. Now, if you had to guess, which part of this do you think ATP would be used in? Cross bridge formation? No, I would, the power stroke?
The grabbing of
The grabbing or the power stroke? It's used completely counterintuitively. So if I tell you that this works like a catapult, then you will understand how ATP is used. When you are setting up a catapult, do you put in the energy by pushing it forward, or when do you actually have to put in energy? To position the catapult, right? To get it into the high energy position. So the place that the ATP is used is to actually get these heads into the point that they actually want to form this and do that. Does that make sense? So let's look now at how ATP is used. We know that when you use ATP, it becomes ADP plus phosphate, right? Yes? Hydrolysis? Okay, so stick with me for a second. We'll go through it one more time. They just changed all this connection today, and now I'm going to kill myself. Okay. All right. So binding of the actin to the myosin, nothing. We don't need any ATP, any ADP, or anything. Now, I'm actually in a high energy position, okay, and now I need to release the catapult. The trigger that releases the catapult is just a phosphate falls off, and the phosphate was from the used ATP last time. We'll go all the way around so you'll understand how it came from. So the phosphate falls off. That's the trigger for the catapult. That's the pin, right? And then you go when you release the phosphate. At or near the end of the time that you've done the power stroke, the ADP falls off. Okay, and I've got an animation for you in just a second. Okay, now I'm here, and I am going to be stuck here unless and until ATP binds for releasing of the catapult and repositioning of the catapult. ATP binds for release, but I'm still not back in high energy position. I can't use my catapult again until I hydrolyze ATP, make it ADP plus phosphate again. Right? And then I'm back at the beginning of the story again, right? Because at the beginning of the story, remember, right before you had your cross-bridge formation, you had an ADP plus a phosphate already bound, right? Right? So that was from when I hydrolyzed it right there to put it back into position. Is that good? Okay. Brady, and then you. Go ahead.
So what you're saying is that when your muscle relaxes is when the ATP gets released?
Not really. I'm not talking about whole muscle yet. So hold on to that question and we will address it. What was your question?
Does power stroke happen once the phosphate is released?
Yes.
Okay.
The phosphate, the power stroke is really, to me, you know how catapults work, right? Put all that energy in to put them like a trebuchet or a catapult, you put all of that energy in, and then as soon as you release the pin, they're flinging, right? So it's the phosphate falling off that's releasing the pin on the catapult. Yeah, and that's when the power stroke occurs. So we're going to go through it one more time with the picture, and then I've got an animation for you, which will solidify it in your head, okay? All right, so tell me what is required. ATP, ADP, phosphate, sticking, falling off when I'm doing something. Okay, cross-bridge formation. Anything? Nothing. Okay, now and I am stuck here until what occurs? Phosphate falls off, and then that starts the power stroke. At or near the end of the power stroke, what falls off? ADP. Okay, now I'm stuck here unless and until I have what? ATP. ATP binds. That allows me to release, and I'm stuck here unless I do what? I have to hydrolyze. I have to use the ATP, right? Okay, I have to hydrolyze it to ADP plus phosphate. I have to put the energy in to get my catapult back into position, right? Does that make sense? And then you start the whole damn thing over again. Okay, so I'm going to show you. Go ahead.
So the ADP and phosphate, at the end when it becomes ATP, the ATP goes away and it changes into ADP and phosphate?
Yeah, so ATP is adenosine triphosphate. When you hydrolyze a phosphate off of it, it becomes adenosine diphosphate, but the phosphate's still hanging out. So it's like I took them apart, but they're both still hanging out on that myosin head. Does that make sense? Okay, all right, so let me show you guys something. I don't know how often you guys use the links page, but because muscle is super visual, there is a complete set of these animations that are set up there for you in order. okay? So if after today and tomorrow you wanted to review all of them, they're all linked for you, but I'm going to go to this one right now because that's what we're talking about right now. And they also have little quizzes at the end, so you can play with the quizzes. They're not my quizzes, but you can play with them. Okay, so what I'm going to see is right here. Okay, we're not ready to talk about calcium, troponin, and everything yet, so let's start right there.
Attached to the exposed binding
Cross-bridge formation.
actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement.
We could do it again.
The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
Phosphate off, power stroke, ADP off, ATP binds, release, hydrolyze, reposition. Constantly, yes. And so to give you an appreciation for how expensive this is one cross bridge, which has to do this multiple times. But now let's go back and look at this little beastie. This is tons of cross bridges. It's 3D. And then remember that there's a bunch of these per cell, and there's thousands of cells per muscle. So you can see why muscle tissue is incredibly energetically expensive. So I don't know who came first, whichever one of you guys.
Hydrolyze happens after the ATP. Right?
No, after ATP falls off?
Is that
You mean hydrolysis is just using water to break that phosphate bond.
okay?
That's all hydrolysis means.
Okay.
So if you hydrolyze the ATP, it uses water to break the last phosphate bond,
That's
right?
Does
That make sense?
The ADP.
To become ADP plus phosphate. Yes.
Okay,
Does that make sense?
Okay.
Is that good? Okay. All right. So go ahead.
So, say you are doing some kind of activity.
Uh-huh.
I don't play soccer, but let's say soccer. And why you are so tired afterwards is because, I don't know, of course, why you're so tired, but thinking of muscles
Mhm.
because you've used so
You've
much
used boatloads of it.
of it.
Yes, and so the more muscle mass you have, the more of this you are going to use. This is one of the ways that you can appreciate that people with a lot of muscle mass, regardless of same body size, are going to have a way higher metabolism if you have more muscle mass. Just so you know, your muscle tissue, after you have your muscle tissue, it's never completely relaxed. It always has partial tone. That's why you can like poke a really hot person while they're sleeping, and they're still like amazingly hot and toned. You should try it, right? I used to be married to one, and now I'm married to my husband. No, I'm just kidding. It's the same dude. Go ahead. We'll get to Charlie horses at the end. We will. Okay. Okay. So you guys are all right with that? Okay. And you can review those with those animations. They're in there for you. And now we're going to add some more detail. So the notes are the length of the sarcomere shortens, but does the length of actin or the length of myosin actually change? What do? They actually slide past one another. Hence the name that describes this, the sliding filament mechanism of muscle contraction. Yes? Now, why is this so damn important? Well, first of all, it is the mechanism of muscle contraction. We can talk about some disorders and pathologies that are occurred with it. And the other thing is by learning this deeply, you get a double benefit in phys because cardiac muscle has almost the same anatomy. It's like skeletal muscle physiology with an asterisk. It works almost the same way. So when you learn this deeply, the cardiovascular system, which comes in the last quarter of the class, becomes way easier, which is important because the last quarter of the class tends to get kind of rushed, right? And everybody's studying for everything, you're kind of chaotic. So this will be a double benefit when you're learning it deeply. Okay, so the objective, of course, is to start at the sarcomere level and then ultimately get the entire damn muscle to contract, right? But you have to start at the sarcomere level before you can understand. Okay, So now we have to back up a little bit and we have to say, okay, I understand what's happening there, but what we haven't talked about is how do we hook this to synapses? Because we already said that a muscle cell will not contract unless it's actually stimulated by the nervous system. So now what we have to do is we have to put the part of the story together in which we understand how synapses work basically, and we understand how sarcomeres contract, but we don't understand the in-between part, okay? But we can do that, right? Okay, so the in-between part is to add in those proteins, troponin and tropomyosin, and then some calcium, and answer the question, how is muscle contraction triggered? How do we do this? Okay, so specifically we are going to use somatic motor neurons, and remember we'll go back to this figure right here, right? In order to get a skeletal muscle to contract, a skeletal muscle fiber to contract, what you're going to have to do is you're going to have to stimulate it with the nervous system. Luckily, this whole nervous system stimulation is the same story we told in the last set of notes. It's basically just a synapse in which the postsynaptic cell is a muscle, right? So the presynaptic and the whole synapse is going to work exactly like you already learned, right? You're going to have voltage-gated calcium channels, you're going to have neurotransmitter release, and you're going to cause a depolarization on the postsynaptic cell. So let's see what happens. So we're going to take this and we're going to blow it up, okay? So this figure I love in your textbook. It's kind of complicated, and we'll also look at it in an animation too. Okay, so how do I get this started? Okay, what are the in-between parts of getting that started? So, in order to get a skeletal muscle fiber to contract, you have to start with nervous system stimulation. So you have to start back on the nervous system, right? So the nervous system portion is, you guys know what has to happen on the nervous system. In order to get neurotransmitter release, you have to have an action potential, yes? And then what kind of channels are involved down here? Sodium up here, but down here? Voltage-gated calcium, and then calcium will move from where to where? Outside into the presynaptic cell, and it will cause exocytosis of whatever neurotransmitter is there. Luckily, the neurotransmitter with skeletal muscle is always the same. We've never found an exception to this. It is always acetylcholine. Okay, so these synapses are always what ergic? They're always cholinergic. As a matter of fact, they're always cholinergic and they're always excitatory because the receptor on skeletal muscle for acetylcholine is always a chemical gated sodium channel. So this is going to be an excitatory cholinergic synapse. Got it? Excitatory cholinergic synapse. Okay. And we haven't found any exceptions to this. I'm not saying they don't exist, but we certainly haven't found them yet. Everybody good with that? Okay. So action potential gets to the end, the calcium channel's open, calcium rushes into the presynaptic cell, causes exocytosis of the acetylcholine. And then what's going to happen is you are going to bind to this portion right here. This portion of the muscle cell membrane, the sarcolemma, that has all of the acetylcholine receptors on it is called the motor end plate. Motor end plate. Anybody want to hazard a guess as to why it's so curvy? Yeah, more surface area. If you have more surface area, you got more room for receptors, and then you can be more immediately responsive. Does that make sense? Yeah? So it's highly curved. It's called the motor end plate. It's got boatloads of acetylcholine receptors, which are also chemical gated sodium channels. And so that motor end plate, so acetylcholine will hit the receptors on the motor end plate. And since they are also sodium channels, it's going to what polarize that muscle cell membrane? Depolarize the muscle cell membrane, exactly. And so the last thing, and then I'm going to show you an animation, and then we'll move on a little bit more. So if you depolarize to threshold, and threshold is slightly different in muscle cells than it is in neurons, but it's the same general idea. If you depolarize to threshold, you'll hit an action potential. Action potentials aren't one way in muscle. They can go from the synapse that way and that way. And then remember that the T tubules allow the action potential to continue down into the muscle. Yeah? So the action potential goes sideways and then it will go down. And what we will see is when the action potential goes down, it triggers a release of calcium from the SR. Calcium gets dumped from the SR. Now let's go back just a little bit before we look at the animation and remind you. Look, the SR is completely wrapped around all of the myofibrils. So if you dump calcium from the SR, aren't you going to dump it onto all of the sarcomeres? Yeah, you're going to bathe the sarcomeres with calcium ions. Okay, so let me show you an animation, and then we'll go back and add just a little bit more detail. Okay, so let's go to neuromuscular junction. Okay, so this is how the story starts, but I don't want to listen to him. Okay.
Hence why they say calcium is good.
Okay, so action potential, voltage-gated calcium channels open. This is still on the neuron, somatic motor neuron. Calcium moves into the presynaptic cell, causes exocytosis of what neurotransmitter? Acetylcholine. And then acetylcholine binds to the chemical gated sodium channels. If you hit threshold, if you depolarize adequately to hit threshold, there are voltage gated sodium channels down here. They're not showing them though. If you hit threshold, you'll get an action potential. So we hit threshold. Okay, so hold that thought for just a second. Okay, everybody go with that? Hit threshold. Now the next, that one's got a little bit more to show you, but now the next animation will show you what happens if you hit threshold. So we hit threshold and it's going down the T-tubules, you see? What do you think this is supposed to be? That's the SR, the sarcoplasmic reticulum, right? And as it goes down, what's going to happen is it will cause calcium release from the sarcoplasmic reticulum. And then we'll pick up right here. The calcium is going to bind to one of those two proteins. So let's go back and pick up those proteins, okay? So there's two regulatory proteins that are on actin. Where are you? Okay, here's what actin looks like before you stimulate it. Notice that you couldn't form a cross bridge if you wanted to, right? That's the default position of actin. And then what calcium does is calcium binds to the switch. I think of it as a switch for the barricade. It is called troponin. When calcium binds to it, it moves the barricade, tropomyosin, out of the way. And then as soon as you move tropomyosin, you can start your cross-bridge cycling, right? As long as you have calcium and ATP, you can continue cross-bridge cycling. Does that make sense? Okay. So this binds and moves tropomyosin, which is the barricade, out of the way. Calcium binds to troponin, which moves tropomyosin out of the way. And then you can continue the story. So go back just a little bit. You see calcium bind. Calcium binds to troponin, the little bitty protein. Flips the switch, moves tropomyosin out of the way. And then you start cross-bridge formation, right? Cross-bridge formation will occur. It's not going to, it didn't add in the ATP, but you know it's in the next animation. And then I get phosphate fall off, and I get my first power stroke, and then ATP falls off, and then I can do the whole cross-bridge formation. So do you see the link here between synaptic transmission and sarcomere contraction? It's primarily calcium release inside the cell, right?
After it releases, does the calcium release so that the
Troponin, tropomyosin?
yeah, tropomyosin can be back in?
Yeah, that's a good question. We're going to have to talk about muscle relaxation in just a second because we haven't talked about how you let go yet. And that's the next thing that we need to deal with. Okay, questions before we add the relaxation part? Are we okay?
This might be completely off topic.
Okay.
The troponin, if somebody has a heart attack, lots of people,
Yeah.
I'm thinking the exact same
hey,
thing.
No, that's a really good clinical question. So when somebody comes in with symptoms that might or might not be a heart attack, right? So sometimes it's just angina, and sometimes it's heartburn, and sometimes it's whatever. One of the things that they can test for is they test your cardiac enzymes. And they're wrong when they call troponin not a cardiac enzyme because it's actually not an enzyme, it's a structural regulatory protein. But what happens is if I have damaged my cardiac muscle cells, this intracellular protein, as the cell dies, gets dumped into the extracellular fluid, and you can pick it up in a blood test. And cardiac troponin is it's the same mechanism. That's what I was just telling you guys. Like once you learn skeletal muscle contraction, you've learned the vast majority of cardiac muscle contraction. There's just a couple of little exceptions, but troponin is part of that. Go ahead. Go
So
ahead.
it's not an enzyme.
It's not an enzyme. And they do call it a cardiac enzyme.
that's all accurate.
Well, they test for other enzymes. I can't remember what the other ones they test for. They test for troponin and a couple of other ones. The other two are enzymes. And this one's it's a cardiac protein, but it's not an enzyme. They're going to argue with you.
Correct them at work.
You go correct them at work. It is it's a structural regulatory protein. It does not have enzyme activity. So there. Okay.
People thinking that-
Okay.
It's always
Um,
on the same day.
and you say neener neener after you say it, okay? Neener neener?
As I walk out the door.
Okay, so basically after we've released calcium from the SR, then you expose the active sites, right? And you do that because calcium binds to where? Troponin, and moves what out of the way? Tropomyosin out of the way, and then you can form a cross bridge, and then you start the whole cross bridge cycling thing, and you will continue the cross bridge cycling thing as long as you have calcium present and ATP present. Yes? Okay, so now to address Brady's question, how the hell do you ever relax this thing? Well, we got to get rid of the calcium, or else you're just going to continue to grab the next cross bridge, right? Okay, so let's go back to the relaxation sequence of events. Just conceptually, does it make sense to you that if we're ever going to try to relax this thing, actually, let me go to this figure. I love this figure, but it's hard to study in class. So if we ever want to relax, we better stop stimulating with the nervous system, right? You got to stop with the action potentials on the nervous system, right? Or else you're going to continue to release acetylcholine. Yes? Okay, you guys with me? Okay, so if you ever want to relax the muscle, you got to stop stimulating it with the nervous system, and that should make sense to you. And then what about the acetylcholine that was there before? What is the mechanism of getting rid of acetylcholine? Well, the mechanism of getting rid of acetylcholine in skeletal muscle fibers is this guy right here, which we stopped before we saw. In skeletal muscle neuromuscular junctions, the way that you get rid of acetylcholine is an enzyme that's present on the muscle cell called acetylcholine esterase. And what it does is it
Now
breaks
to
acetylcholine.
acetic acid and choline.
It breaks it down to acetic acid and choline. And then it reuptakes the most valuable part, which is choline, right? And it's called acetylcholine esterase. Say that, please. So if you want to remember how to spell it, it's just acetylcholine with a st and then an erase at the end, because that's what it does. It erases acetylcholine. Acetylcholine st erase. Go ahead. It is on the motor end plate. Very good. Okay, so it's on the motor end plate. So you guys are all right with that? So that is the way that you get rid of the neurotransmitter that was already released. Okay, so let's say we stopped stimulating it, we got rid of what was already released, but now what else do we have to do to relax the thing? In order to relax the thing, in addition, we are also going to have to take care of the calcium, correct? Because as long as the calcium is present, then you're just going to grab the next cross bridge, right? So the way that I get rid of the calcium is to put it back where I got it. Where did I get it from? Sarcoplasmic reticulum, good, the SR. And the sarcoplasmic reticulum stores boatloads of calcium. So to get it to be released from there, you can just release it by diffusion. But to get it to go back in, you're going to have to use what? Yeah, you're going to have to pump it. Yes. You're going to have to pump it against its concentration gradient. That means, although it seems counterintuitive, muscle relaxation is an active process. Yes? It does use ATP. In order to get the damn thing to relax, you're going to have to pump the calcium back in. And then in addition, remember that you can't actually relax the muscle. Remember, show me where we get stuck if we don't have any ATP. So we go cross bridge formation, that's fine. Power stroke, that's fine. I'm stuck here in the absence of ATP, correct? Okay. So in order to get that to release, I also need ATP. Does that make sense? Okay. So you need to pump the calcium back in, and of course you need that last ATP to get the cross bridge to release as well. Okay. So as far as relaxation goes, you have to stop stimulating with the nervous system. You have to get rid of the acetylcholine that was already released with acetylcholine esterase. And then you have to pump the calcium back into the SR for storage. And then with all that removed, your troponin will go, the switch will go back down and the tropomyosin will be back in the way, and then you will release. And by the way, the relaxation occurs relatively passively because look at these little springy proteins. Those are called titin. As soon as you let go, it'll just spring back into position and back into its relaxed position. Does that make sense to you guys? Okay, so questions? And then I'll talk about some disorders and we'll reinforce this.
Can you do it just one more time?
Okay,
Tell us what.
what was the question?
I don't know.
I don't ever remember what I say,
I
or else I probably wouldn't say it, honestly.
have a sarcomere contracts. Can you do it one more time?
Okay, you guys do it with me. Everybody get up here and get flapping. Everybody get up here and get flapping. Don't hit your neighbor. Okay, ready? Ready? What is this? What is this? What is this? Myosin head. What's it reaching for? Actin active site. When it binds, it's called a cross bridge. Cross bridge formation. Now I want a power stroke, and I won't get one unless what occurs? Nope. Phosphate falls off, and then I get a power stroke. Right? We all look really cool. You get a power stroke. Now I'm stuck here until what occurs? Wait, actually something just occurred at the end of the power stroke. ATP fell off. Now I'm stuck here unless what occurs? ATP binds, but doesn't hydrolase, and that allows me to do this. And now I'm stuck here and can't use my my catapult again until what? Hydrolysis. ATP goes to ADP plus phosphate. Yes? Right?
So where's cross bridge in that? Where's cross bridge in?
Cross bridge is as soon as the actin active site and the myosin head are bound. That's cross bridge formation. So it's when you reach up and grab the handle. Yeah.
And then power stroke is.
Power stroke is this.
It's telling you if you make a little
What?
ground.
Do you make a little diagram? Yeah, you got your whole body for a diagram. You just go flopping around. I'm gonna watch you guys during the test, and you're gonna be flopping. What? Sure, just don't hit your neighbor. I don't care what you do. And nobody's looking at you either. Anyway, it's just like yoga, right? If you're looking at other people, you're gonna fall over.
No, girl, you can't do that.
Okay, so now let's bring this home and talk about the problem with being dead. The problem with being dead. There's probably a lot of problems. Actually, there's very few problems with being dead. It's probably the end of problems. The problem with being dead is that you don't have any ATP. And you don't have any ATP primarily because you run out of what? Oxygen. Exactly. Glucose is not generally a problem. You usually have a decent amount of that stored, but you run out of oxygen, so aerobic cellular respiration is something that you cannot do. And in the absence of oxygen, remember, you're going to have some problems letting your muscles let go. And it's going to be a progressive thing. So what's going to happen is in the absence of ATP, remember, you can't pump your calcium back into your SR, correct? In addition to not being able to pump your calcium back into your SR, calcium will be leaking out of your SR, right, down its concentration gradient. So you're slowly getting calcium buildup, right? And when you get your calcium buildup, all of your myosin heads that can will find the next available cross bridge, right? And then they can do the power stroke for free, right? Okay? They can't do 20 power strokes for free, but they can do 1 power stroke for free. And then they get stuck right there. And then you can't release once you run out of ATP. So what happens is not immediately after you die, but a few hours, and it's temperature dependent, right? So if you're in the Texas heat, it's all going to happen real fast. If you're in Minnesota, it's all going to happen real slow. But what's going to happen is that slowly, unless somebody takes you and sticks you in a fridge or somebody starts to preserve your body, you are going to get a progressive leaking of calcium and a buildup of muscle contraction. And it's not going to be like wiggly muscle contraction. Like if this guy probably died flat on his back, right? And you get a progressive tightening of muscles, right? And so the muscles will tighten. Now look at how hard that would be to do if you were alive. I don't even know. There's probably one person in this room who can do that.
Me.
When she's alive.
Oh, what?
No, I think it's Elizabeth.
Oh.
Who could probably do this. But even she would be going, take a picture! Take it now! So this is muscle tightening that you have. It's not generally going to change your position very much, but it will make it very difficult to move you around, right? Especially if you don't die laying flat on your back. So one of the reasons that I decided that I was going to pay attention to academics and get a college education is because I did not want to die in a trailer in Texas like half of my redneck family, okay? So I'm telling you, this is my big motivation. I'm like, I'm going to eventually own a home, and I'm not going to die in a trailer, right? And I've accomplished, the rest of my life is gravy after this point. Because those little big trailer bathrooms, right? You've been living wrong. You're dying in a trailer in the heat of Texas, and then nobody finds you for 72 hours because, you don't have anybody who likes you. And then they try to come get you out of the bathroom, and your muscles do not let go, and you are in this position, right? And you haven't eaten your fiber, so you died taking a dump. That's like a bunch of people die on the toilet. A bunch of people. It's really sad. You should eat some fiber. That vasovagal response when you're bearing down. Wait. And then what happens is you die like this, and you got this little bitty bathroom, and then you're also probably fat. I mean, let's just be serious. Then they have to try to get you out, right? And you don't get out. We're being honest about the South today.
Honest.
We're being honest, right?
Mm-hmm.
And they have to get you out, but your muscles won't let go. So what are their choices at that point? What do they do? They can break your bones or break the wall, because they got to get you out of there before you get to the squishy stage. And that, my friends, is why you need a college education. Glad I could help. Brady, did you have a comment?
Well, I was just gonna say, when my brother died and my sister died, he was like leaning up against the wall and like he was stiff.
Mm-hmm.
So he has done that for a while.
Yeah.
But he was on the ground, and the only thing that like was messed up was like his neck. Because his neck he fell.
Oh yeah.
And so it was
So his body was stuck in that position. Yeah. So with rigor, how quickly do you go through it? Well, it's temperature dependent. Like all of your proteins are going to degenerate and degrade more quickly at higher temperatures and more slowly at lower temperatures. When are you going to go out of rigor? When your proteins start to denature. When your proteins start to fall apart, then the actin and the myosin can't hold on to each other anymore, right? You're losing the structural integrity of your proteins. So you go floppy after you die, and then you get stiff as a board, and then you go floppy again, right? And then eventually you go squishy and liquid. So something to look forward to. Okay. So that is, I don't know where this picture came from. I'm pretty sure I'm not supposed to have it, just so you know.
I
But
was thinking, gosh, that is a terrible way to die. Like,
I don't think he died that way.
Like, could you imagine?
You probably
Someone picked him up.
Someone picked him up and posed him and then sent me the picture.
My God, I would hate to die.
No, if he had died, his butt would be on the ground.
Naked like that. That would be freaking
What?
No, he didn't die that way. Somebody got in trouble. That's a med school, like somebody got in trouble picture or morgue picture. Okay, so now let's talk about something, I don't know. So let's talk about polio, also called poliomyelitis. So polio, I'm going to tell you what happens, and then you're going to tell me the impact on the muscle, right? Because you're going to understand the muscle physiology. Polio is a virus that's almost completely eradicated from the world. There's a couple of little pockets in Africa and Asia where polio pops up every once in a while. It's almost been completely eradicated. But the virus actually destroys somatic motor neurons. It kills them. And you know that neurons tend to be amitotic, right? So if you destroy them, you're kind of screwed. Okay? So what is going to be the impact on the muscle tissue if you destroy the motor neuron? What's it going to do?
it's gonna make it
Temporary paralysis, permanent paralysis, what? Permanent paralysis. It's going to cause permanent paralysis. And the impact of the permanent paralysis will be eventually you're going to start to lose your muscle tissue, correct? Because muscle tissue is very energetically expensive to maintain. And then after you don't use your muscle tissue, you will start to degenerate your bone tissue, right? Because you need to maintain your bone mass by wiggling your bones. And so this causes a progressive deterioration and permanent paralysis. So remember when you got polio? No, you don't, because vaccinations. That's why you didn't get polio. So you aren't even vaccinated against polio anymore because there was such an effective vaccination campaign. And my parents and your parents and grandparents' generation that polio has almost been completely eradicated. Every once in a while it pops up. I am living in fear that it is going to pop up in some yoga community in San Francisco because they think the green juice will take care of polio, right? No, vaccination works really well, and this is a deadly disease, of course, because this is a skeletal muscle, right? And if you have complete paralysis of the diaphragm, then you lose your capacity for respiration. What about partial paralysis of the diaphragm? Anybody knows what happened with polio patients that had partial diaphragm paralysis? Iron lung, have you ever heard of iron lung? An iron lung changes the pressure, so it increases the air pressure so that you push air in, and then it releases it so that you can release the air back out. And so you can live in an iron lung partially or completely. We had a president who had polio. Do you know who that was?
FDR.
FDR had polio. He had partial paralysis because of polio. He, however, was astute, and he knew that partial paralysis, he wasn't photographed in his wheelchair, never with heads of state in his wheelchair. He was always standing with other heads of state in his wheelchair. People,
That is, whatever, on his legs.
yeah, he had his braces on his legs, and he was ambulatory, but not for very long periods of time. So that's polio or poliomyelitis, and it causes permanent paralysis because of loss of the motor neuron. What?
So it like deteriorates your muscle.
It deteriorates, but do you remember when we talked in anatomy, bone mass needs to be maintained by stress, right?
Yeah.
And if you stop stressing your bones, you'll actually start losing your bone mass. Just like astronauts in space start doing that, right? So what happens is you lose the muscle mass and then eventually you lose the bone mass. I mean, you're not going to lose your bones completely. You're just going to lose a lot of bone mass, right? So that's polio. A couple other things. Let's talk about cramps. What is a cramp? From a physiological perspective, I'm not talking about uterine cramps or menstrual cramps. I'm talking about skeletal muscle cramps. What happens is usually because of an ion imbalance like hypokalemia or hyponatremia or something, you can have action potentials that fire on the motor neurons inappropriately and therefore can cause like painful tetanic contractions on skeletal muscle. Like anybody volunteer for the muscle zapping last week in lab? You did? Did it get to the point that it hurt at all? Oh, no? So you can get to the point that you're making like tetanic contractions and it really hurts. So hypocalcemic tetany is specifically tetanic contractions because of low levels of calcium. Remember, calcium is your latch on your sodium channels. If you lose it, then you get a bunch of inappropriate action potentials and you can get into tetanic seizures. That's usually because of a hormone imbalance. Muscular dystrophy. Have you guys heard of muscular dystrophy? Okay, muscular dystrophy. There's several different kinds. I'm going to talk about one of the common kinds. Muscular dystrophy is a genetic disorder. It's the one I'm thinking of is X-linked recessive, so it's more likely to happen in which gender? Males, exactly, because you've only got one X chromosome. In order for a female to get muscular dystrophy, she would have to have a double dose of this mutated gene on an X chromosome, which is unlikely unless my mom, dad, or brother and sister, or I just had the worst luck in the history of the world. Okay, so what happens is this mutation causes an error in a cytoskeletal protein, so that the cytoskeleton of cardiac and skeletal muscle is weak. But remember, when you contract, what do you pull on? You pull on the cytoskeleton, right? And so what happens is it causes a progressive deterioration of skeletal and cardiac muscle. How do you think that exercise helps or doesn't? No, it doesn't help at all. Exercise causes a greater and more quick deterioration. The problem is that you can't stop using your diaphragm and you can't stop using your heart, right? So what generally happens, and it's usually little boys, is that they are fine until they're supposed to start walking, and then their gait and their pull-up is a little weird, and then they figure out that they've got muscular dystrophy, and then they have this sort of progressive deterioration, and usually they die of cardiac or respiratory failure, because you can't stop using them, right? And there isn't a cure. There are some genetic treatments they can do.
Same as Lou Gehrig's disease.
It's got similarities, but this one's genetic and that one's not.
Is this the one where you can tell if your child's about 2 and they can't
Yes.
stand? They're like
Mhm.
pulling.
So there's this weird, I can't remember what the gait pattern is. There's like a typical way that like you've either been a baby or you've had a baby. There's a typical way that kids generally like pull up. and these kids have really weak musculature in their legs, and so they do this. They have a harder time like supporting their body with their legs. So that's usually how the first diagnosis occurs. So anyway, that's muscular dystrophy. This one's an autoimmune disorder. Autoimmune disorder, I'm going to tell you what it does. You tell me how it impacts the muscle. Autoimmune disorder, your immune system makes a mistake and it starts to destroy your motor end plates. What's going to happen to the muscle fibers?
It's not doing anything.
Motor end plates. What's going to happen?
Acetylcholine.
Yeah, you can't stimulate it. So that muscle fiber is paralyzed. It's not permanent paralysis because you don't destroy the cell. You destroy the motor end plate, and if your immune system leaves it alone, you'll regenerate the receptors in the motor end plate. So what it tends to manifest is like Bell's palsy, right? You have like the facial paralysis, and then that'll come back, and you'll be fine, and then you'll have weakness here, and then you'll have trouble breathing, and then you'll have it in your legs, and it doesn't go away. It just sort of roves around the body. It can be deadly because of the respiratory problems. So that's myasthenia gravis. Tetanus and botulism have a lot to do with one another. Tetanus is caused by a toxin produced by the bacterium called Clostridium tetani. And botulism is produced by a bacterium that is similar, same genus, different species, Clostridium botulinum. So each one of these, when they get into anaerobic conditions, what do I mean by that? Anaerobic conditions. No oxygen. You get them into anaerobic conditions, they will start to produce a toxin, okay? And the toxin is what gets you. It's not actually the living bacterium. So with tetanus, how's your mom tell you get tetanus? Step on a rusty nail. And as always, mom is at least partially right, okay? So the problem, it doesn't have to be a nail. It doesn't have to be rusty, okay? And it doesn't have to be your foot. What it has to be is a puncture wound that closes on itself, right? Because then it creates an anaerobic condition. Does that make sense? As it closes on itself. Now, these bacteria are really, really ubiquitous. They're all over the damn place. But they only cause a problem when you get them into an anaerobic condition, and then they start to produce this toxin. It causes a problem with neurotransmission at skeletal muscle neuromuscular junctions, and it can cause like lockjaw. How'd your mom tell you get botulism? Dented cans, and she was part right, as usual, right? This one is, so when you're like, let's say you're home canning something, right? And you're home canning your green beans, and what are you supposed to do to the jar? Boil the hell out of it. What are you supposed to do to the lid? Boil the hell out of it, right? And then you're supposed to make sure that the stuff inside is sterile, and then you were supposed to vacuum seal it. What if you skipped one of the steps? You might have organisms that are still living, and then you're going to create an anaerobic condition with the vacuum, right? And then what happens is if the organisms are still alive, and it was Clostridium botulinum, it'll start to produce the toxin. And then you're like, oh, well, I just cooked my green beans. The toxin is thermostable, right? It's really, really stable. And so what is the whole dented can thing? Well, if the organisms are still alive in there, they're going to produce carbon dioxide as a byproduct, right? And so that little dimple that's supposed to be pushed down, pushed up, don't eat it, right? If you open the damn thing and it hisses at you for half an hour, don't eat it, right? You know that. Shh. That is, you're gonna die. It's gonna be terrible. Don't feed it to your dog either. They've got the same skeletal muscle problem. So let's talk about Botox, because now we figured out how to make this work to our advantage. Botox cosmetic is the fact that we figured out how to make the botulinum toxin in the laboratory. And since we can make it in the laboratory, we can use it therapeutically or cosmetically, whichever. So with Botox cosmetic, what happens is if you can't do this, you don't get these, right? Right? So all you have to do is shoot some in right here, right? And it will temporarily destroy your synapses at your skeletal muscle for about 4 months. And then your wrinkles will slowly go away, and you will never look surprised or excited or anything. So that's what happens there. What?
Just pull your neck back.
No, it does not work. I tried.
Stick some tape up here, right here. Right here. So that's how Botox works. It has been used therapeutically, however, and it's they use it for hyperhidrosis, like people who have like so much sweating. You can destroy the neurotransmission there. They can use it for migraines.
And they're
Yeah.
not doing it for feet for like plantar fascia issues. And
Really?
Yeah, they were, because I had that, and they were like, "We can stick something." I'm like, "No, you can stick some Botox right here, not in my feet. They'll see my feet."
FYI, if you do decide to get Botox, and I know we're like all, when we're in public, we're like, hell no, and I'm like, oh, maybe. Go to somebody who's been to med school and knows where the damn nerves are. Don't go to a wine party in which the nurse is drinking with you. That's not a good idea. Pay a little more for it so you don't end up all cockeyed. So,
It's like,
what?
It only lasts for 4 months though.
it only lasts for 4 months, but it's got a progressive impact if you do it more frequently. Girls are starting to do it as young as 17.
I know.
So that they
All the
never
nurses
get
I work with
wrinkles.
do it, and they said, "Nikki, have you ever thought about doing it?" I said, "I will stab you in your sleep. Don't talk about it."
I earned all
I
this.
earned all this.
So that's Botox cosmetic, and that's like how it works. It's kind of interesting. They actually do, interestingly, when people have facial either nerve damage or muscle damage, if you go to a plastic surgeon who's really, really well trained, they can use really tiny Botox injections to make your face more symmetrical. Because they know what the hell they're doing. They're like, this one's just slightly, so if like you had some kind of nerve damage that made this eyelid droop, well, what if I actually like make your face symmetrical by actually making the other side look the same? Or you can make the muscles, and muscles are arranged in antagonistic groups. And if you paralyze the antagonist, then sometimes it will actually come up. So, but that's like med school.
Those are the really pricey.
Yeah, those are pricey. Yeah, for sure. Okay. Last but not least, before you guys leave, and then I'll tell you what we're going to do for next time, is sometimes there are chemicals that affect the neuromuscular junction. When we talked about curare before, but now that we know muscle, I want to talk about it again. Then I want to talk about nerve gas, and then I'll talk about what you guys are going to do for next time. So curare is that eat the monkey out of the tree without climbing the tree drug. What it does is curare is an acetylcholine antagonist at skeletal muscle neuromuscular junctions. So what's going to be the impact of curare on the muscle? Is it going to contract, or is it going to be paralyzed before it contracts? Which one?
Paralyzed.
Yeah, it's going to be paralyzed. So it's called flaccid paralysis, in which like the monkey can't contract its diaphragm, so it falls out of the tree, right? Or its muscles. But the other one, is nerve gases. Also some pesticides, but we invented nerve gases to do this. And I know that we're in the habit of thinking in the United States that we all have like a stellar human rights record, but I would like you to know that most of the nerve gases were actually invented by the United States before they were actually picked up by other countries. And one of the nerve gases, one of the biggest categories, works like this. It blocks the impact of acetylcholine esterase. So tell me what it would do to the muscle. Blocks the impact of acetylcholine esterase. You can't relax. So if curare kills you because you can't inhale, right? You can't inhale in your diaphragm. This one kills you because you can't exhale.
Oh, jeez.
Oh.
That's what those nerve gases do. Also, some pesticides weren't designed to do that, but that's the impact they have on humans.
Who was
So-
it that was killed over in China?
His brother, stepbrother.
Yeah, stepbrother.
Because of this,
Two
they
women ran up to
sprayed
him.
it.
One had one, and the other had the other, and it was
A nerve gas that was
Nerve-
too
Two
nerve
nerve
nervous.
gases,
thing
they
and
kind of
killed him.
sprayed it.
When did this happen?
over the weekend.
Over the weekend.
Oh, it was new, too.
I've been off social media ever since the election, so I'm blissfully ignorant. I've also read 6 books in February, so I highly recommend being off social media. Although I have, honestly, I've read 2 nonfiction books and 4 novels in February. I've written 3 abstracts, and done 2 scientific presentations and read 5 articles. So I highly recommend getting off of social media. So let me tell you what's happening for. So before Wednesday, here's what I'm going to do. I'm going to upload a worksheet for you guys. And this worksheet will be for us as a class to practice muscle contraction and relaxation. It's going to be a fill-in-the-blank worksheet. And what we're going to do is you're going to bring it, and then we are going to go through it as a class. I'm going to call on you individually to answer some of the blanks. And I don't care about success or failure. I care about being engaged in the process, okay? Because it's actually going to be to review this. So you're going to have to figure out what's supposed to happen on the presynaptic cell, and then what, and then what, and then what, and then what. And we'll go all the way from the neuron stimulating it all the way to get to skeletal muscle relaxation, okay? So I'll post it for you later today, and you'll bring it to class, and we'll do it at the beginning. We won't have a quiz until a week from today, okay? See you guys on Wednesday.
Not to cause problems, but about the lab, should we write that stuff down now?
You just send it to me today, tomorrow? Yeah, it's fine. No, you're not causing problems. We're trying to fix the problem.
Right.
So who got killed? Who was it?
The stepbrother of Kim Jong-il
Yeah,
from Red China?
stepbrother, he ordered like his two and to they watched you play the airport, sprayed here, and we and then did you like
Whoa!
Yeah, he ran up to security and said, I think I've been poisoned, and he was dead like that.
Wow, that's some old school spy stuff, man. Why couldn't you have sent him in a car?
And because they had to have both parts, thus the women weren't injured at all, because they only had one part.
So was it like aerosol?
I heard it was on their hands, but, I don't know.
So what if you're with a system? Like if you're building a flat and you increase your muscle mass, but then you start working out, so would it just be more movement? I'm kind of thinking like yo-yo that are like yo-yo exercises.
Next time, maybe I should write a little bit of the physiology on that.
I'm curious because so many people do like binges of training.
Yes.
And so I'm wondering if it's usually for things like if you're not going to make a lifestyle change, it's just kind of better not to